Hello and welcome everyone to this video podcast for Human Genetics. In this video podcast, we're going to continue our discussion of what happens when chromosomes are not maintained properly. More precisely, we're going to talk about the mechanism that leads to aneuploids, and then we're going to talk about 4 different kinds of chromosomal structural rearrangement. So let's go ahead and get started. Now I want to spend just a little bit of time talking about how aneuploids form. We already said it usually results due to egg development during oogenesis. But don't forget, it still can occur due to problems during sperm development. Now, I'm not going to go into great detail about how aneuploids form, but you should be aware of non-disjunction. And this is the unequal segregation of chromosomes during meiosis. I'm going to focus on meiosis right now, but on the next whiteboard, I'm going to also explain how non-disjunction can happen during mitosis. Remember, meiosis produces oocytes and sperm, and mitosis produces new genetically identical cells. But we'll come back to that in a moment. So we know that in meiosis, normally, you have your chromosomes that enter it that have been copied. And I'm only going to show one chromosome. But remember, while this is happening, all 23 pairs of chromosomes are doing this exact same thing. And usually what happens is, during meiosis 1, you produce cells that look like this, where these two get segregated away from each other. These two homologs get separated away from each other, and you just have the cells now with the sister chromatids in them. And then each of these will go through meiosis 2, where the sister chromatids get pulled apart. And then these are your gametes. So remember, this was meiosis 1, and this was meiosis 2. This is what usually happens, or is what is supposed to happen. Non-disjunction is when these chromosomes fail to pull apart. So if they fail to pull apart, and they fail to pull apart because, and again, we don't need to worry about the details, but during meiosis they stick together. But they should unseparate, and sometimes they don't. So you could end up with a situation like this, where both of these homologs, both of the pairs of sister chromatids here, don't separate, and they end up in the same cell, and nothing in this one. Now remember, it's not really nothing because the other 22 pairs of chromosomes did segregate properly. When this happens, it's not universal and every single chromosome sticks together. It's usually just one of them. And then usually what happens is they fall apart here. They're no longer stuck to each other. And the rest of meiosis works fine. These two sister chromatids here, they pull apart and go into each cell. These two sister chromatids pull apart and go into each cell. Now this is where the problem is, because now let's just say this is chromosome 21. And usually, the whiteboard I had before, it only had one copy of chromosome 21 in it. Now, because of non-disjunction, you have 2 copies of chromosome 21. And if this were to be fertilized with another gamete that did not undergo non-disjunction, and so it only had one chromosome in it, one chromosome 21. It ran out of room here, but when they come together during fertilization, they would produce a cell with 3 copies of chromosome 21 in it, or an individual who would have Down syndrome. Non-disjunction creates gametes that will ultimately lead to a trisomic individual, or they end up producing a child that is a monosomy. And that would happen on this path down here, where in this case chromosome 21 all went into this one gamete, and no chromosome 21 went in here. And so it's going to end up producing gametes with no chromosome 21. If one of these gametes here is fertilized with an opposing gamete that has one chromosome 21 in it, through fertilization, it's going to result in an individual, a zygote, with just one copy of chromosome 21 in it. And a monosomy 21 is going to be lethal. Remember, all monosomies of autosomes are lethal. The only monosomy that survives is the X chromosome monosomy. Non-disjunction can also occur in meiosis II, but don't worry about that. Your book talks about it. I just want you to know this general pathway. Now I want to talk about non-disjunction occurring during mitosis. Let's make this chromosome 18, the one that would lead to Edwards syndrome. And let's say this egg here had one copy of chromosome 18, and then this sperm here also had one copy of chromosome 18 in it. And they would form a zygote after fertilization. And at fertilization, they're going to be a euploid. They'll have two copies of every chromosome, including chromosome 18. This is what usually happens. But sometimes what happens is after the zygote forms, it'll divide like it should. It'll produce 2 cells. It'll produce 4 cells. It may produce 8 cells. At some point early in development, what happens is mitotic non-disjunction occurs. And you have unequal distribution of chromosomes in this early embryo. And let's just say it's this one here. This one ends up with 3 copies of 18. All the others have 2. Remember, if it had 3 copies of 18 at fertilization, then that zygote probably wouldn't even make it this far. And if it did and made it to birth, that child would probably die during their first year of life. But in this case, what happens is you have 1/8 of the cells that are trisomic. And how that translates as this embryo develops into a fetus and then ultimately into a live birth, you have an individual born then that is approximately 7/8 euploid and 1/8 trisomy 18. And we call this, and this is a term I said before, I just never defined it, so I'll define it now. This is what we call a mosaic. Since this individual is mostly a euploid, in most of their cells they have the correct chromosomal content. And in 1/8 of their cells, roughly, this isn't exact, but about 1/8 of their cells, they have trisomy 18. Because of that, they typically are healthier than I'll say pure trisomy 18 child. I put quotes around healthier because they're not healthy. They still have many of the symptoms that someone with trisomy 18 might have, but they're not as severe. And if you remember, I said that when I talked about trisomy 18, trisomy 13, I said sometimes they live a few years. And in some cases, they've lived 13, 18, 20 years. And in all of those cases, it was because they were mosaic. Because most of their cells were euploid, yet some of them were still of the trisomy 18 variety. So mosaics appear in a person as a mixture of euploid and some trisomy, and in some cases some monosomies. Now, if you remember, we began this discussion a couple podcasts ago about the structure of a chromosome. And then we moved into what happens when there are problems with the chromosome. And we said there are 2 problems we should be concerned about. One was the number of chromosomes, which were the aneuploids and the polyploids, which we just talked about. But we also said at that time that we had to think about chromosomal rearrangements. And there are 4 chromosomal rearrangements that you need to know. And we're not going to go into quite as much detail as we did with the chromosomal numbers, the aneuploids and polyploids, but you should know these basic things: deletions, duplications, translocations, and inversions. Some of these may appear kind of obvious what they are, but be careful because all of these only concern part of the chromosome. So for instance, let's talk about deletions first. If you had a chromosome like this, and you had genes on it, A, B, C, D, E, F, a chromosomal number change would be to maybe make 2 copies of the whole chromosome, or delete one of these through a monosomy. But deletions, in this sense, means that we're only deleting part of the chromosome. So we may delete that, just genes E and F. And because of that, usually deletions are not as severe as a monosomy. Now, if you delete the wrong genes, yeah, they can be lethal. but you can delete some of these genes. Now it's typically more than 1 or 2 genes. Usually it's a fairly long region, and so the chances of some kind of problem associated is still there. Now before I go on with deletions, I'm going to put duplications next to it because I think there are some similarities between these that we can cover at the same time. So again, let's draw our chromosome. And remember, this is what it would normally look like. A duplication might be something like this, where we've duplicated one part of this chromosome. And usually it's not 1 or 2 genes. Usually it's a significant portion of it where there's many genes involved. So on the surface, just like when we talked about aneuploids, we might not think there's a problem with this because we're only increasing the number of genes by 1 or decreasing it by 1. And so we might not think that's a problem. But if you remember from our aneuploid discussion, sometimes it can be a problem. We have the genomic imprinting phenomenon, and I won't go into great details about this again. You can go back and watch that. But just remember that it's possible that the genes we deleted here were the only ones being expressed, and the ones that are remained were genomically imprinted to be off because they came from mom specifically or came from dad specifically. We also have a numbers balance issue, just like we talked about with the trisomies. It's possible in this case here that A and B, when they're made, interact with another protein, we'll call it Z, and this does a job, some kind of function. But now, if we have 2 copies, an additional copy of A and B, maybe they bind to each other in such a way that now prevents Z from binding to it, and now that will lead to no function. Often, the proteins we make are made in a very precise amount, and messing up with that amount can have problems. The only other problem we should mention with deletions is that deleting a centromere leads to a monosomy. If we remove this centromere, let's say we removed all of that, including the centromere, now when the rest of this chromosome goes through meiosis or mitosis, without that centromere, it won't get segregated properly during mitosis or meiosis. That's all you need to know about deletions and duplications. Alright, now let's talk about translocations. And with translocations, let's draw 2 chromosomes. We'll draw chromosome 14 and let's draw chromosome 21. This is what we typically see in a human cell. Now sometimes, due to a translocation, one of these chromosomes, or sometimes it's just part, but let's talk about the whole chromosome ending up on one of these chromosome 14s. And the mechanism we don't need to worry about so much, but the result is this, or could be this, where you have 1 copy of chromosome 14, 1 copy of chromosome 21, and then this translocated copy of 14 attached to 21. So in this individual, let's just put a circle around it so we know that this is a cell, and this could be 1 cell amongst the trillions of cells in that person. This person still has 2 copies of chromosome 14 and 2 copies of chromosome 21. And so you might say, well, Gorsuch, that person should be fine. And if you were to say that, you would be correct. This person is usually fine. The problem comes in the next generation. And the next generation, based upon how these chromosomes can now align, because there is some, for lack of a better way of saying it, some confusion on how these chromosomes are going to line up during meiosis. You can end up with some different possibilities. For instance, you could end up with a gamete that looks like this, where you end up with the translocated chromosome 1421 and the chromosome 21 all by itself. Now this is just one possibility. I'm not going to exhaust this list and show every possible combination from this, but I want you to focus on this one here, because when this cell here, it could be an egg or a sperm, but when it is fertilized with another cell that has just 1 copy of chromosome 14 and just 1 copy of chromosome 21, we end up with a cell that looks like this. This zygote that forms from these 2 gametes has 2 copies of 14 in green, but it has 3 copies of 21. 1, 2, 3. And so that is what we would call Down syndrome. It doesn't form due to a non-disjunction. It forms due to an inheritance of this translocated copy. So we call this kind of Down syndrome, it still has the same phenotypes, the same consequences, but we say it's inherited. Non-disjunction trisomies are not inherited. There'd be no reason to expect that if one person had a trisomic child, that they would have another trisomic child due to a non-disjunction event. However, this person here, with this translocated copy of 1421, will likely have more than one child who has Down syndrome. So if you were to see a problem that said something on the lines of, there was this family and there were 8 kids and 3 of them had Down syndrome, what's the likely cause of it? The likely cause is that there is a translocation of that person with a chromosome 21. Often it's 14, but it doesn't have to be, but it is often 14. It would not be because of just some really bad luck and they're constantly getting these non-disjunction events. Now let's talk about this last one here, inversions. Inversions are when you have your chromosome here, and for this case I'm just going to draw one of them, A, B, C, D, E, F. An inversion occurs when part of the chromosome is cut on both ends. In fact, let's add a couple genes here, G and H. So it's cut on this end, in this case between D, E, and F, and then it's flipped. So you end up with a chromosome that looks like this, where DEF has been flipped to FED. And like with a lot of these, we look at this and say, well, there's probably no problem because you still have genes DEF and FED, they're just in a different order. And sometimes there are no effects, but some problems that can occur with these inversions are the place where it's cut can be in the middle of a gene. So it's possible, I drew it right at the boundaries of the gene, but when these happen, the events don't know where the genes are. They just randomly cut the DNA. So you could have cut right in the middle of F or right in the middle of D. And so that's a problem. You could also, in a similar way, remove promoters, depending on where it gets cut. And if you don't have the promoter, you can't turn those genes on. So that could be a problem. Another problem is something we call position effect. And this is a serious problem, or potentially a serious problem. And I don't want to go into enormous detail, but what I do want to tell you is that in the centromere region, there's poor expression of genes. So in this example, there's reason to believe that gene D would still be expressed, but it would be expressed at a lower level. Gene F, further away from the centromere, would be expressed at a higher level. And so when we flip them, we've now placed gene F, assuming that we haven't cut it in the middle of the gene or we haven't removed its promoter or anything like that, but we've placed gene F at a place on the chromosome where there's just naturally less transcription. And we place gene D in a region that is now more expressed. And there might be, through evolutionary selection, a reason why we went low expression of D and higher expression of F. And by flipping this around, we've changed how these genes might be expressed. The last thing, I'm just going to list this one, I'm not going to explain how it happens, but a lot of times people with inversions can have fertility problems. It has to do with how things get balanced during meiosis, but in general, I just want you to know that there can be fertility problems with people who have inversions. Okay, that's all I'm going to say about chromosomal rearrangements. All right, the last thing I want to talk about in our discussions here in these podcasts about chromosomes is uniparental disomy. This is when you inherit 2 chromosomes from one parent and not a chromosome from the other parent. So for instance, we know a person could get 2 copies of 21, 2 copies of 21 from, say, dad, and then 1 copy of 21 from mom. And yeah, they did receive 2 copies from dad. That's not what we're talking about. We're talking about an individual who's born and they have 2 copies of chromosome 21 from dad or from mom, and nothing from the other parent. All the other chromosomes, you have 1 from mom and 1 from dad, but for 1 chromosome, it comes from 1 parent. And it doesn't have to be chromosome 21, I'm just showing that one in this example. This is really rare though. So for this to happen, well, a few ways this could happen, but one way it could happen is you could have a sperm with no chromosome 21. And then you could have an egg that was formed that has 2 chromosome 21s. And as a result, they would have a child with 2 chromosome 21s, both from mom, none from dad. Another way this could happen is you could have a zygote that has 3 copies of 21. 2 that are of a paternal origin, and the other, say, from a maternal origin. So this is a trisomic individual, and if it's chromosome 21, they would have Down syndrome. But you can get this uniparental disomy is because sometimes early during embryonic development, very early in embryonic development, one of these chromosome 21s is recognized as being dangerous and it's removed. And if you happen to remove mom's, then you only have 2 copies from dad. And so that would be an example of uniparental disomy. And again, I've been focusing on chromosome 21, and I think in the examples I gave, I said you have dad with 2 copies. It can go either way, and it can be with any chromosome. Again, might think at first surface that this isn't a problem because you still have 2 copies of chromosome 21, but one of the big problems that we have to think about here is our good friend that's coming back to revisit us every now and then, and that is genomic imprinting. So because of genomic imprinting, we know that some genes that dad gives us are expressed, and those genes are not expressed in mom, and vice versa. Some genes that dad gives us are not expressed, and they're expressed in mom. If we have 2 copies from one parent, then we lose that balance, and that can cause problems. And this is one of the ways that either Prader-Willi, that we've talked about before, or Angelman syndrome can form. That's all you need to know about uniparental disomy. And this does bring us to the end of this rather large discussion of chromosomes and how they're tested and how they're detected and what happens when various abnormalities in copy number or structure occur. If you have any questions at all, please make sure you contact me. If not, I'll see you on another podcast. Bye for now.